I noticed in order for New York to get a handle on this, actually the governor of New York went ahead and said private labs can test, as opposed to the initial protocol, which involved only state and federal labs, right?
Yes. Yeah.
Why was that? And why was that a necessary step? Or like what we're seeing now, which is the state of Maryland, the governor went ahead and secured 500,000 tests from South Korea.
Yeah.
Why is that happening in the United States?
You know, I think that's a case of governors really wanting to do the best for their people and their citizens. And they're listening to their public health experts and their physicians and their scientists, and they're seeing the need for testing. And if I can't get the brand of bread that I like to get at one supermarket, I go to another and I get that brand of bread, or I get that item that I need. And if I can't get it from one shop, I go to another shop. And I would really like to see a robust federal leadership on this with clear guidelines on these tests are going to be the ones we use, and these are the ones that are our backups, and we've got enough testing for everybody, but we don't have that. So you're seeing this patchwork quilt being developed of tests coming in from South Korea to Maryland, or a commercial lab that's stepping up and giving tests to through their commercial sites, their storefronts in certain regions. And, you know, by hook or crook, we're going to get the testing that we need. It's just coming together, like I said, in more of a patchwork network rather than a unified response.
So now, validation, I know you studied testing validation a lot. How do we validate all these different entities, or is that not a concern here?
You know, that is a concern, because you don't want this to turn into the wild, wild west. You know, you don't want me and my mom cooking up our own test and then selling it on eBay or Amazon because that again is irresponsible, because you're going to have false negatives and false positives. Yeah. So this goes back to, there's two entities that I really enjoy reading their literature from. One is the FDA, and they have an ability to kind of waive some of these fastidious testing guidelines that they have to put the tests through. The tests themselves have to be tested before they're rolled out into the general public.
Right.
So they have the ability to do what's called an EUA, emergency use authorization. That is a quick sample test. If it does all right, then they approve it with an EUA and it gets rolled out to the public. There's also another accrediting or regulatory body called CLIA.
Yes.
This is a great group. These people look at what kind of tests are suitable to be performed where and how accurate these tests are. So, you know, you've got CLIA, which is clinical laboratory information analysis, I believe, or analytics.
I'm CLIA lab director.
There you go. I'm sorry, I don't know the name. I should. We just call it CLIA. But you've got CLIA that really accredits all these tests and these bodies, and they say this test is an easier test to do, perform, and interpret. So you don't need as robust a training, meaning you can have more lay people administer the tests, which obviously makes it get out into the community easier. And then at the highest level on the CLIA, you've got to have, you know, board certified pathologists that run a very strict, tight lab and all these regulations and inspections. So you've got this gambit in between those levels. And I think they do a wonderful job of really demonstrating to people the levels of the tests, who can perform them, and they validate. And their stamp means a lot to me. I think they do good work.
So basically what you're saying is, if someone, maybe they're a private practice doctor or they run a surgery center and they're thinking of doing serology, one of the things they should consider is looking at CLIA's list of approved tests or CLIA endorsed tests as a way forward.
Absolutely. I think if you're going to do it, you want to go with people that have already validated your test. You want to get what you pay for. You don't want to get a knockoff test that, you know, is just Kool-Aid and a swab. You want to get what you're paying for. Your patients deserve this. The community deserves this. So I think they do a great job. And, you know, I was on their website earlier today looking, and they've got a laundry list of approved tests, manufacturers, where to buy them and how to get them. And that's awesome. The FDA has something similar to that. The CDC has something similar to this that really is bringing the ability for that family doctor, that private practice physician, to reach out and say, all right, I'm going to buy some tests and I'm going to start testing my people in my panel. And I think that's awesome.
That's awesome. And I think that's important because I feel like that's one of the concerns I keep hearing from a lot of my colleagues, which is I really want to know more. But I feel like, especially when it comes to serology, what you're hearing from a lot of doctors out there is how do I know which antibody test to choose? And what do I do with that information? And how do I find the information? Because there are a lot of companies already doing direct marketing to doctors. And practices who are saying, use our test, use this, use that. But of course, as everyday clinicians who aren't studying epidemiology the way you do, their worry is, how do I know I'm picking the right test? Or it may be someone in a country far away who's also interested, no science background, but the public health interest is there. And they're wondering, how do I go about finding tests that we can use? And so I think this is really important information. And I'm glad you're able to discuss it with us, because that's so important. So you're saying go to the website, they even tell you the skill level that's needed.
Yes, it's wonderful. You can go, you can visit, and you can look at, they're called CLIA waived, for humble doctors like myself. These are the tests that we typically do, in an outpatient clinic or things like that, point of care at the bedside or in the clinic. And these are going to be meeting their stringent standards. And like I said, it's threatening to get into the Wild West, where you have these tests that are out there that haven't been evaluated by these accrediting bodies. And I think with that, you have a real caveat emptor, buyer beware, because, the worst thing you can do to your patient is perform a test on them that has no clinical significance.
Right.
You've put the patient at risk of a test, you've taken time, you've taken money, and you don't have any actionable information usable at the end of your exam and your testing period. So that, I think, it's better to do nothing than use an improper test. And I think that the guidelines of where to get those good tests can be found on that website, definitely. Yes, ma'am.
I agree with you. And you just mentioned something that a lot of people are also interested in, which is that point of care or rapid testing. What's that? And is that different from serology and the NAAT tests? And how so? Yeah,
so these are, these are your I call them RADTs, rapid antigen detection tests. Most everybody has probably seen this. I think probably the most common one is that rapid strep swab that we do so much of in our pediatric populations. You go back, you tickle the tonsils, and then you get the classic plus or minus, the yes or the no. You get similar to a pregnancy test. So these look at antigens. Antigens are what is signaling your body to attack something. So this is kind of like putting a laser to guide your immune system to attack this. You're looking for the antigens released by this virus, and you're testing those. Now, the World Health Organization has been clear that they're not on board with using these clinically yet. They are advising these for research purposes only, just because I think they're trying to validate them with the NAATs and the serology testing, kind of bookending the problem and then finalizing the validity on this. So if you have the rapid antigen detection test is the game changer. That is the swab, 5 minutes later, plus or minus, and you're good to go. But these are also sometimes the most complex because there is no gray area. It's either the black or the white. But that's a holy grail.
Isn't one of those already approved in the US? What is that test, the EUA, that got an EUA about a month ago?
Yeah. So this is approved and it's being used right now. My take on this is that the specificity and sensitivity isn't there yet, which is why, again, I side with the World Health Organization that you need to do that two-step confirmation, or you need to do it in conjunction with a research protocol, meaning you are a university or a medical system, and you're doing your test, but then you're backstopping it with one that has a higher rate of true positives or false negatives that can confirm, yes, it's there. And then you report this data back, because the people that are manufacturing these tests, the scientists, those virologists, those immunologists, they're always tweaking this test and trying to build that better tomato. So they need this information, and it's not there yet. It's close. I'm hoping in the next month or two, we're gonna get some more EUAs coming out, but I don't believe it's there yet as a mono test alone.
And that's the thing about oftentimes proteins or carbohydrates being produced from the virus. As the virus mutates, these protein structure can mutate, and their amino acid makeup can mutate or change, and that can change the sensitivity and specificity. So I agree with you. I wanted to hear your take on it, because that's been my take on the rapid point of care, and why for me in my clinic I have declined bringing it in, because even though it's available to some who are CLIA directors, I don't see how I can use it and be relying on it, especially not just for telling my patients they're negative, but for me as the doctor, being sure that my patient is negative and I'm not now exposing myself because of a test that I did that said it was negative.
Exactly. You know, it's a holy grail, and it would be a big game changer. I don't think the technology's there. I liken it to, riding on a horse and trying to throw something into a bucket on another moving horse. So there's a lot of coordination that has to happen, and it's coming. It's going to be there. I'm very confident in the scientists. It's just I don't think it's quite there yet. That's why those nucleic acid amplifications and the serologies are going to be your two big players right now. But those are your bookends, like I said, to help get that rapid antigen test developed.
So for now, if you guys are listening, what Dr. Eric is saying, the nasopharyngeal swab is going to be here for a while yet, right? So you don't get to show up and tell them, I don't want that one, do something else. If you really want to know if you're positive or negative, you have the best chance of knowing from the nasopharyngeal swab.
Yes,
the first time around.